All right, so you guys ready to rock and roll with 17.1? See behind me, the sun is starting to come up. All right, so let's go. So 17.1 is about genes and variations. Our goal is to describe how mutation and genetic recombination increases genetic variation. Okay. All right, so far side's kind of cute, kind of cute. Class abruptly stopped practicing. Here is an opportunity not to only employ the skills, but also save the entire town. You guys, I haven't even had my coffee. I don't even drink coffee. I haven't had my tea yet. That's probably what I need. All right, so our 17.1 objectives is to define evolution in genetic terms, identify the main sources of genetic variation of population, and also state what determines the number of phenotypes for a trait. So let's think about it. Here we are, this guy again, you recognize him, Darwin. Darwin developed his theory of evolution without knowing how inheritable traits pass from one generation to the next, or whether heritable variation came from. Where did it come from? What would happen when genetics answered questions about heredity, how heredity works? So, define evolution in genetic terms. Let's go. Researchers discovered that heritable traits are controlled by genes. Changes in genes and chromosomes generate variation. For example, all these children receive their genes from the same parents, but they all look different. An organism's genotype is the particular combinations of alleles it carries. An individual's genotype, together with environmental conditions, produces the phenotype. We learned about that before. Phenotype includes all physical, physiological, and behavioral characteristics of an organism. Natural selection acts directly on phenotype, not genotype. Some individuals have phenotypes that are better suited to the environment than others, and these individuals produce more offspring and pass on more copies of their genes to the next generation. So a population is a group of individuals of the same species that mate and produce offspring. We learned about that before. A gene pool consists of all the genes including all the different alleles for each gene that are present in a population. Researchers study gene pools by examining the relative frequency of an allele. The relative frequency of alleles is the number of times a particular allele occurs in a gene pool compared to the number of times other alleles for the same gene occur. For example, this diagram shows the gene pool for, no it doesn't, fur color in a mice population. Forget that. Evolution is any change in the relative frequency of alleles in the gene pool of a population over time. Got the wrong picture, sorry. Natural selection operates on individuals, but resulting changes in allele frequencies show up in populations. That's important to understand. Populations, rather than individuals, evolve, which proves Lamarck. Remember that guy with the giraffe in the neck? Proves him wrong. There you go. Oh, there's the mice one. I didn't click over. Sorry about that. There's your mice thing. Give me a couple minutes to look at that. Seconds. All right, objective 1 is define evolution in genetic terms. So in genetic terms, evolution is any change in the relative frequency of alleles in the gene pool of a population over time. Objective 2 is identify the main source of genetic variation in population. Dun, dun, dun. Mutations. Mutations that produce changes in phenotype may or may not affect the fitness. Some mutations may be lethal or may lower fitness. Others may be helpful, beneficial. People are born with roughly 300 mutations, you're all mutants, that make parts of our DNA different from that of our parents. Most are neutral, a few can be harmful, and a few are beneficial too. Mutations matter in evolution only if they can be passed from generation to generation. So you may have a mutation but doesn't get passed on, then or because maybe you don't reproduce, it doesn't matter. Mutations must occur in the germline cells. Those are the ones that go through meiosis because they produce either the eggs or the sperm. So, most inheritable differences are due not to mutations but to genetic recombination during sexual reproduction. Meiosis again. Each chromosome in a pair moves independently during meiosis. Remember, you get some of your moms and your dads in this cell and some of your moms and your dads in that cell. So in humans who have 23 pairs of chromosomes, this process can produce 8.4 million gene combinations. Crossing over, tetrads, which occurs during meiosis, another way in which genes are recombined. Crossing over further increases the number of new genotypes created. Genetic recombination is the reason why individual members of a species differ from one another. Lateral gene transfer, I should say, occurs when organisms pass genes from one individual to another that is not its offspring. This can occur between organisms of the same species or organisms of different species. Lateral gene transfer can increase genetic variation in the species that picks up the new genes. Remember we were talking way long ago about bacterial transformation? They can do it. Remember that scientist who did that? What was his name? Oh gosh, it was Avery and Griffin. Yeah, okay. All right, so objective 2. Identify the main sources of genetic variation in the population. The 3 sources of genetic variation are mutation, genetic recombination during sexual reproduction, and lateral gene transfer. There you go. Objective 3. State what determines the number of phenotypes for a trait. A single gene trait is a trait controlled by only one gene. Single gene traits may have just 2 or 3 distinct phenotypes. The most common form of the allele can be dominant or recessive. Dominance of an allele for a single gene trait does not necessarily mean that that dominant phenotype will always appear with greater frequency in the population. Remember when we did the assignment with the dimples? Dimples are dominants, but when you did the whole class, pulled the class, you found that most people did not have dimples. Another example of a single gene trait, oh, I didn't click on, I'm so sorry, is the presence of dark bands that appear on the shells of a certain species of snails. Even though the allele for shells without bands is dominant, a population may show a greater frequency of the white bands phenotype. Polygenic traits are traits controlled by 2 or more genes. We learned that too. Each gene of a polygenic trait often has 2 or more alleles. What is a good example of a polygenic trait? You remember? Skin color. A single polygenic trait often has many possible genotypes and even more different phenotypes. Human height, which varies from very short to very tall, is an example of a polygenic trait also. The bell-shaped curve in the graph is a typical polygenic trait graph that you might see. The frequency of the phenotype. So objective 3 is state what determines the number of phenotypes for a trait. The number of phenotypes produced for a trait depends on how many genes control the trait. So to recap here, 1, our objectives define evolution in genetic terms, identify the main sources of genetic variation in population, and state what determines the number of phenotypes or trait. Make sure you can do that because they all support a goal of describe how mutation and genetic recombination increases genetic variation. Ooh, flowers this time. Enjoy.